Hi, I'm Jonathan Keats from the Translational Genomics Institute at the Myeloma 2022 meeting here in Scottsdale, Arizona. And we just finished presenting some of the data on the update from the Multiple Myeloma Research Foundation CoMMpass study, which really has been, I'd argue, a transformative study for the field because it took a different model of trying to collect a very large cohort, uniformly characterized molecularly, but with serial data collection that increases month on month. And we're now just getting into the last year of this study, so it was a nice opportunity to present to the field how the study has evolved with time, and it's been used by so many people taking this model of not holding the data until one research group published it to actually making it available to the community so the community could rapidly evolve how we view the underlying disease. And I think it's been a very transformative data set for the field to build on. One of the things that we talked about here was just starting to understand what is really important in myeloma when we talk about high risk, because we have certain features, I think, coming out of this data set have led us to understand what is like ultra high risk, so patients who have lost both copies of a common tumor suppressor called P53 is really one of those groups of patients, though it's really small, maybe only 5%. This is a group of patients we know we're not really doing a good job for today. And then we have other things that we talked about from a high risk, so gains of a whole chromosome arm, the largest chromosome in the human genome, chromosome 1. When we see this, we've started to appreciate in the field that this is an important prognostic variable, but it has some weird underlying features that some patients have the whole arm but only one extra copy. Some patients have the whole arm but 2 extra copies, and we think having 2 is more important than having 1. And then we also have another small group of patients who, instead of having the whole arm gain, have little focal gains on 1q that only include 10 or 100 genes. And those tend to have very high gains. And trying to sort out what the biology is of those different features is very difficult unless you have a data set like CoMMpass where we have 1,000 patients and all those different molecular measurements, so we can subset patients into those groups. And I think it's those kind of observations of helping the field now understand what's there and then bringing that updated clinical data so we actually really do understand how it interacts with the therapies received and how, regardless of therapy received, how it impacts a patient's long-term outcome. And I think these are things that are going to lead to huge improvements in the field.